arrived to the last part of this morning, and we have heard a lot about the molecular mechanism and an overview on the possible application of different laser techniques. And I'm very happy that Bettina has spoken about a lot again of the molecular mechanism of soft tissue laser, because we have covered the possible application of lasers from soft tissue, from soft lasers, from soft tissue, from soft lasers to very high power lasers. And we have arrived, we have finished with a discussion on application of ionizing radiation and possible techniques. We have mentioned photon and electron acceleration by linear accelerators. Is there anyone who remember on the dose rate, the maximum dose rate, which we could achieve with linear acceleration technique for photons? 10 rate per minute, yes. And now it is important to remember, and we can apply very sophisticated multi-field irradiation with large integral dose, but a very homogeneous and very selective radiation delivery to the target. And we have spoken about proton therapy, and we have seen that with protons, with charged ions, we could achieve much more selectivity, and even with carbon ions with higher biological effectivity, when we are not depending so strongly on oxygen and oxygen supply, because the direct effects are dominating. And if it is so good, it is so beneficial, why it is not used on a wider range? Actually, up to now, you remember that around 15 million person has cancer, a new cancer diagnosed yearly, and 2/3, 75% of this, no, 3/4, okay, so 75% of these patients receive radiotherapy. So the radiotherapy is applied more than 10 million people yearly, and you can see that altogether less than 1% of this patient receives hadron-therapy, any form of hadron-therapy, proton therapy or carbon magnetic. Why? Because it is extremely expensive. A part of America, there is one center or two centers in France, some centers, 4 centers in Germany, some two in Italy. So very few centers, and even less centers which provide carbon ion beam as well as. And the reason of it, there are 2 centers in Japan, and the reason of it is the extreme high cost of the construction and maintenance and operation. And there are a lot of research devoted to somehow decrease the cost, to make more compact, smaller, because the other problem is the high cost, and the other problem is size. It is like a mountain. You can see that only the cyclotron part diameter is 25 meters. So it is really this cyclotron, where this eye therapy was performed, what I have shown you, it is really built in the middle of a mountain, one mountain. And this large size is not proper to build to a hospital area, because the whole hospitals will be occupied by one cyclotron. So the size should be decreased and the cost should be decreased. There are different methods. The compact cyclotron and the success of it resulted in recently the last 20 building of cyclotrons in Germany, for example, which is already really embedded into a medical area, but still quite large. So you're still on a pavilion base and not filling the building. But the fixed field alternating gradient dielectric wavelengths, they are all approaches, and what we are speaking about, the laser-based approach, the plasma wave field technique, and the so-called tabletop laser technique for charged particle therapy, which should be energy and intensity modulated, compact proton or ion facilities. And meanwhile, the development of and building the ideas how to produce protons and ions, it turned out that there are several advantages of laser-driven photons, and with laser technique, very high energy electrons could be generated as well as. And that is an emerging interesting aspect, the effect of infrared femtosecond laser pulse filamentation shown to be beneficial as well as in cancer therapy. I shall show you that. And the laser accelerated photons and electrons has advantages, the ultra intense beam, and this is what I have mentioned, that this is 10 to minus 10 or even less second the dose delivery could be done, the ultra short dose delivery, because of the very high dose rate, and it will allow intraoperative radiotherapy, special achievement of special beneficial because of a very short pulse duration, and this very short duration can lead to repetition of processes which is shorter than the molecular processes. And there are some indications that this will be cause less damage for the healthy tissues and no change for the tumor tissues, because the tumor cells have less capacity for repair, and the repair mechanisms are intact and well working in the healthy cells. And if we can produce irradiation within very short pulses, it may be give us a larger difference between the response of tumor cells and cancer cells. And the other thing, that they can be, the laser-induced beams can be produced in a very small beam length with higher resolution of intensity modulation than the normal therapy. So already the photon and electron therapy can be produced with some advantages if it is done on a laser, with a laser-based technique, and it is not large because according to this publication, and if I see the therapy, you will have a lab visit today or tomorrow. You will see that it is really not large. Some tables put together, and the laser system, high power laser system, can be operated on that. So 100 mega electron volts can be achieved with a small system. And in this talk, I try to summarize where the world is actually with the development of such like high power lasers and what is going on. There were a lot of theoretical work devoted prior to start the experiments to how to make particle acceleration with lasers, on beam design. There are a lot of mathematical calculation of dose distribution of the generated beams, and there should be a lot of measurement on those as well as. So we have spoken about that practically this very high power laser it destroys the normal construction of the atoms and separate the electrons and the nucleus, the positive nucleus, and this charge separation, if they are accelerated, result in charged particle beams. I have already told it previously. The plasma formation is the major action of this high power laser, and the electron cloud of the negatively charged electron cloud takes the fast positive ions with them, depending on the energy, and the laser energy of some 1,000 joule, and very short pulse duration, high intensity, and the repetition rate. This ELI apps will have a very high repetition rate, can be improved reasonably recently to provide us the hope to get medically useful proton and carbon ion beams. The main procedures to accelerate ions, the target normal sheet acceleration, where the hot electrons generated, and electrostatic field takes the ions and protons from the plasma with a certain kind of energy. And the other mechanism is the radiation pressure acceleration, where the high radiation pressure limit the thermal process, and the radiation pressure and the electrostatic force balance result in the charge particle separation. And this is the two main action, the target, two main procedure, and there are a lot of theoretical work still going on. In our room, when we are situated in the Eli Alps provisional building now, then theoretical physicists are calculating which is the best method to generate the medically useful energy range protons, target normal sheet acceleration, or the RPE, the hole boring method, which result in the pressure of the target, which is a thin foil, and what I wanted to mention, then the electrons, the very high energy electrons, are generated in gas target, but the protons in a very thin solid target, and the circular polarization inhibits the hot electron generation and switch the regime of TNSA to RPE. This was published already 1 decade ago. It started in 2000, in this century. The design of medically useful accelerated protons and carbon ions production with laser-based technique, with full optical system, or in combination with radio frequency acceleration after the generation of the beam. And the first proton acceleration experiments at 3 different centers, because there is a competition, of course, was published in 2000 already, and 60 MeV proton energy has been reached, but it was not stable. It could be not produced during hours long. It was one record, and that's it. So we have recorded the record, and there are a lot of further consideration on development of technique, how useful beam can be developed. And the main design is that the laser system is positioned in a certain room, and in the nearby room the laser light is LED, and both fixed particle beam and rotating gantry can be somehow designed. Of course, there are a lot of aspects and parameters which should be designed. The beam, the particle should be correctly selected, the fluence selection, the intensity should be properly tailored, the beam transport should be solved, energy selection system should be built in, energy modulation, because a range of energy, changeable energy range is needed, and there are a lot of theoretical thinking about the different dose delivery methods, the spot-based delivery, the lateral layer-based delivery, axial lateral-based delivery, or combination of all of them. And this is, if we go closer a little bit, here is the last focusing mirror and the target for plasma generation, and from this target the released ionizing beam, which goes through a system which provides us the possibility to select the energy, first the particle and thereafter the energy of that. There are quadrupole magnets which make a beam and a certain size of the beam, and the fluence, the intensity selection system, further magnets and dipole pair magnets, and everywhere diagnostic devices or dosimetry devices built in order to control the system. Of course, the radiation protection aspects should be solved. This is a plan for radiation protection within the head of the potential laser-driven accelerated beam, and this is the room design. Of course, thick shielding for radio protection purpose, and there are already achievements with large energy carbon ions and ion accelerations. The first biological experiments has been started as soon as the first beam was provided for radiobiologists, and it turned out that a very intense, very high dose density photon beams, laser accelerated photon beams, laser accelerated electrons, protons, carbon ions, at present did not show any major difference from the point of view of biological effectiveness. If they apply in a pulse or a continuous mode, because this experiment in Dresden was looking for the difference in between the continuous and pulse mode irradiation with cell cultures using colony farming assay as biological endpoint, and gamma-2 H staining, which is staining for the double DNA damages, double strand break DNA damage. And the other type of research is done on a theoretical basis with a potential beam of laser plasma generated very high energy electrons. It means some hundred of mega electron volt, but for example at LIFs giga electron volt energy could be achieved on the basis of the present calculations. And the Monte Carlo simulation for radiation treatment of the prostate resulted a little bit surprised, because up to that point, because it was not available, very high energy electrons were not available, nobody have calculated a dose distribution, and it was very similar, which could be achieved with protons, with other positively charged particles. So this 150, 250 MeV energy electrons proved to be very advantageous from a lot of aspects. For example, in this beam have a small diameter, so these beamlets provide us a much better intensity modulation, and the dose distribution is really comparable with proton dose distribution. And this is a very interesting emerging thing which was detected, that the irradiation with directly femtosecond pulse duration, very short duration laser, cause different effect in tissues, in water, than the larger pulse duration beam. And the shorter is the pulse duration, starting from femtosecond range, so some tenth of femtosecond or attosecond, even attosecond, the majority of the energy is transformed not to coagulation or something like this, but not evaporation, but all of the energy is transformed to ionizing radiation. So it forms a plasma within the water or within the human body, and it goes even to deeper side because of this filamentation effect. I am looking always to look if I tell something silly, but he should correct me. And already animal experiments had been performed after, of course, careful dosimetry, and with this chemical dosimetry in water and in water equivalent gel material, they could show the ionizing effect of the laser filamentation of the laser beam, and thereafter they started to irradiate tumors which were enucleated. So this was a tumor xenograph in a nude mice on both legs. Here is the control, which was not treated, and this part was treated with 5 separate spots of 100 to 300 micrometer of diameter during 10 minutes, and it was irradiated during 2 weeks, and the tumor of 6 to 8 millimeter like this was completely diminished, completely destroyed. And if it's true, it will provide us a fantastic tool in our fight against cancer. In our therapeutic palette will be supplemented with a method which provides no entrance dose and no dose behind our target, and our tumor can be very selectively treated. Of course, there is a lot of work to really work out all of the details to establish this method really for cancer therapy. We have to look for what is happening with deep-seated tumors, what is the filamentation effect, the attosecond pulses, what has happened, what will happen with opaque and scattering tissue, and what is happening if pigmented or hemoglobin containing tissue has a differential absorption of this type. But nevertheless, it is interesting and encouraging that it may be one way to go on. And the proton beam development with laser technique, proton acceleration, were experimentally as well as provided for radiobiology groups. So the first experiments were started and below 10 megaelectron-volt, which provide a very low penetrance, and breaking is occurring very close to the surface. So, but it was proper already to irradiate different tumor cell types in a cell culture, and the gamma H2AX double-centric staining and the 53EP1 staining did not show any difference in comparison to the normal cyclotron-generated proton source and laser-induced protons. And this shows how much work should be done prior to an experiment, because the whole beam production, proton beam production, takes place in vacuum. So first there should be a solution for get out the beam from the vacuum, to get rid of the elements, the particles which is not required, to provide an energy range which is stable, and to place the samples and the dosimeter system somehow. The best is if at the same time, in real time, those measurements could be achieved to the beam. And of course, never only one sample should be irradiated. This is a campaign where a lot of samples should be irradiated, and the beam which is provided should be stable, and the whole experiment repetitional, reproducible. And there were a special device, I don't have the photo on it, which is like a small elevator which was elevating all of the small pieces of cell cultures to the beam with high accuracy, of course, to expose exactly each one to the beam, and then to go down to the shielded space, and the whole experiment was like this, automatically performed together with an integrated dosimetry and cell irradiation system. The Faraday cup and the film dosimetry system was built together with a flask of tissue culture containing flask. This experiment was used around 10 to 15 MeV protons in Dresden. Meanwhile, in Munich, they could achieve a little bit higher energy protons that they have irradiated as well as cells, detecting the relative biological effectivity, and they have irradiated animals, small mammalians like this mouse, and this mouse has an ear tumor, and in his ear tumor cells were inoculated. Do you know why an ear? Because it's thin enough for these low energy beams. And this, the starting was after 1 year, lots of work to validate this experiment, and the proper positioning device was, as it's worked out, at first with electrons at Jena. And the human head and neck tumor xenograph was irradiated in the leg of the mouse, as you have seen previously, with 23 megareclamer protons. It was already proper for that, this higher energy, and no difference in RBE, relative biological effectivity, could be detected. So up to now, we can see that the laser-driven ionizing radiation, from a biological point of view, very comparable. We could not detect any major difference between the cyclotron generated particles, which is logic. The only thing is high dose rate and the pulse and the continuous mode difference, but it's shown no real biological difference up to now. And this high power infrared laser may give us an interesting opportunity for research and for going further on that way. And if the ELI apps will be built here, Szeged, it will work together with the other pillars of ELI. ELI Med in Prague is dedicated especially for biomedical research, but it was a lot of question why we would like to perform as well as biological research. We would like to perform a certain type of biological research, but I think that if there will be 100 laser-driven radiation centers, everyone could have his own dedicated research field. So there's a lot of aspects which could be detected and scientifically experimented with this ultra-short pulses, ultra-high dose rate, and in ELI apps with high repetition rate. We would like to work with photons, very high energy electrons, which will be provided earlier than proton and carbon ions, and we would like to try the attosecond pulses for this filamentation effects, on the way that if all of this laser system will be installed and the secondary sources built, then we try to help to the facility design, to the transport system, dosimetry, dose calculation, radiation protection aspects, and finally the biological experiments can start on with irradiation, dose escalation, fractionation, in vitro and in vivo biological system using different endpoints. Our group worked together, or included the molecular biological aspects as well as a part of other changes. And Monte Carlo calculation has been already started for optimizing the beam parameters. We're working on dosimetry research to define the proper detector. We would like to use polymerizing gel and gel dosimetry especially for this laser filamentation, but all of the other effects. And of course, it has to validate in comparison of other detectors in different phantoms and in comparing with the calculation for for the aim of offline measurements, and we would like to establish online dosimetry as well as. Actually, the standard in laser-induced proton sources or hadron beams are these radiochromic field image plates, microchannel plates, Thomson spectrometers, and so on, which is listed here, and we would like to add our geodosimetry system to that. And our biological experiments will include cell cultures, spheroid cultures, and especially fishes, cebradianial small fish embryos seems to be very proper for such like purpose. We have started already 5 years ago a lot of experiments with ionization radiation, with neutron sources, with different radiobiological effectivity sources to establish the proper setup for biological experiment at ELI labs, and thereafter animal experiments can be done as well as, to the aim finally to work for our children or grandchildren to have a laser-based clinic, a laser-based cancer center, there from the screening to the surgical techniques, the laser evaporation, surgery, and laser-based diagnostic led to earlier detection of cancer and more appropriate diagnostic provide as early cancers for effective therapy with laser-based irradiation systems. Thank you very much for last slide I was not able to put in the system, so I thank you for your attention with the ELI apps design.
So thanks for that. How new is this discovery using femtofilamentation in parallel?
Some years, it was published some years ago, and the majority of the scientific society does not believe. So it was a surprise for us. Really, you have looked for the literature in details, but I have found only two or three articles, and it was so encouraging, and there was no follow-up, nothing. It was first Congress abstract, and thereafter this journal article was quite reviewed journal and accepted journal, but everybody is very uncertain regarding this.
Yeah, because there's been so much development in femtosecond 800 nanometer lasers, they are so available.
Mm-hmm.
So that's a good card for ELI.
Yes, I think so. Even we try to get access to Tevati system, which actually exists and which can operate with some tenth of 30 femtosecond pulse duration. But to build up an experimental setup, it needs so many little details, and the success of an outcome of an experiment is really depending on the good definition and well design of these little details. So it took, for example, in the Haselier in Germany, more than 2 years to establish this one experimental setup for laser-induced proton beam. It's easy to tell, but it is not standard at all, so we have established all of the parameters.